My name is Stanford Mabasa. I'm going to talk about essential microelements or micronutrients which are required by the plant. Initially, we talked about macronutrients from water and air, which are carbon dioxide, which are carbon, hydrogen, and oxygen. Although we didn't do much on this, otherwise we talked a lot on the macronutrients from the soils. That is nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur. Today we are going to concentrate on micronutrients, boron, chlorine, manganese, copper, iron, nickel, zinc. I will display them in the next slide. So we've got manganese, molybedenum, chlorine, zinc, boron, iron, copper, and nickel, which is not shown here. Here we've got a list of 8 macronutrients. They are required in smaller amounts. We've got iron, manganese, boron, zinc, copper, molybedenum, chlorine, nickel. We're going to start with the iron. This element is added in several forms. For example, it can be added as ferrous sulfate, F++, or ferric chloride, F+++. Iron is added as iron chelate. Iron chelate is a metal atom bound to an organic compound by 2 or more bonds forming a ring structure, e.g. sesquiterene. Please note that the latter is expensive but the best source of iron. pH is critical for the iron availability. A solution that is too basic, that is above 8, can cause problems in iron uptake. The best pH range is 5.6 to 6.6. Here we've got chelated iron. Then here we've got a plant where, I think a crop where iron was treated, where iron was applied, and an area where iron was not applied. You can see very short plants here. I've got also 2 plants, petunia and barley. Petunia acquires its iron in the ferrous state, whereas barley acquires its iron in the ferric state. Now let us focus on the overall functions of iron. Iron is involved in enzyme activation as a catalyst in redox reactions and electron transfer, and it acts as an oxygen carrier. It is involved as an enzyme activator or cofactor in the synthesis of chlorophyll and in the functioning of several other enzymes, including catalase, peroxidase, ferredoxin, and the cytochromes. Please note that ferredoxin and the cytochromes contain iron. It is therefore crucial for chlorophyll production, protein synthesis, and respiration. In legume plants, iron is important in nitrogen fixation. It also participates in the terminal step of respiratory chain in other hemi-enzyme catalase and peroxidase. For example, catalase can activate the reaction from hydrogen peroxide to water and oxygen. Iron deficiency and toxicity. Now, iron deficiency is experienced in soils containing a lot of calcium with the high pH, where lime-induced chlorosis is observed. Since iron is usually bound, chelated to various compounds in the plant, it is fairly immobile. So we expect to see symptoms on the young leaves, where we see interveinal chlorosis, like what is shown on these pictures. Toxicity symptoms, these are not usually seen. However, if foliar applications of iron are used in excess, necrotic dead spots on the leaves may appear. Iron toxicity is the second most limiting factor in crop yield in wetland rice, where flooded conditions enhance iron reduction from ferric to ferrous state, increasing its availability to toxic levels. Manganese is another element. If it is deficient, we can see white patches with green veins. Toxicity symptoms manifest themselves in the form of brown spots. These are precipitates of manganese in the older foliage. Sometimes chlorosis, and even chlorophyll distribution may be evident. High manganese levels may cause iron deficiency. There is usually an overall reduction of growth. Now let us focus on the functions of manganese. It activates enzymes in the following processes: carbohydrate metabolism, phosphorylation reactions, citric acid cycle, and Hill reaction in photosynthesis. In a Hill reaction, water is split into oxygen and hydrogen. Boron in the solution, in the soil solution, occurs as H3BO3 or BCH3. It is best taken up from solution that is below about pH 6.5. The overall functions of boron. Boron is related to the metabolism of calcium and potassium. It is used to regulate carbohydrate metabolism. It is involved in RNA synthesis. In addition, it is involved in lignin biosynthesis, where it participates in diester and cis-diol configurations of lignin precursors. The boron requirement is much higher for reproductive growth than for the vegetative growth in most plant species. Boron increases flower production and retention. Elongation, germination, and seed and fruit development. It is involved in plant hormone regulation. Boron plays an important role in regulating hormone levels in plants. Plant hormones like animal hormones regulate many growth and reproductive functions. For example, flower initiation, fruit development, cell wall and tissue formation, and the root elongation are all influenced by hormones. A deficiency of boron can cause incomplete pollination of maize or prevent maximum pod set in soybeans, for example. Boron deficiency. Boron deficiency is a very common nutritional disorder because under high rainfall, boron hydroxide is readily reached and the boron availability decreases with increasing soil pH, that is liming. Boron is not mobile within the plant, and younger growth shows symptoms first, just like what happens with iron. With the boron deficiency, shoots show abnormal or retarded growth, and then blackening, and both shoots and roots tend to die back. Stems and petioles may be brittle and develop cracks. Young leaves may appear thick and curled. Flowering and fruiting are restricted or inhibited. Typical rots of fruits and vegetables may be attributable to boron deficiency. Terminal buds and the youngest leaves become discolored and may die. You can see these spots showing boron deficiency. Now, with boron deficiency, internodes are shorter, giving plants a standard rosette appearance. Intervernal chlorosis and mature leaves occur. Flowers, buds, and developing fruits, borers for cotton, typically drop, severely reducing economic yield. Generally reduced seed and fruit set are common, with the growth and quality of fruits severely affected. For example, internal cork in apples and a decrease in the pulp peel ratio in citrus fruits. In vegetables, in vegetable crops, e.g. lettuce, water-soaked areas on the leaves or on the tips, which become brown, black. Now, talking on boron toxicity. Boron toxicity is associated with use of municipal composite and arid, semi-arid conditions where irrigation water may contain high levels of boron. Typical symptoms on mature leaves are marginal or typical chlorosis or both. Necrosis. Toxicity symptoms may appear as deficiency. Now, zinc is absorbed by plant roots as a zinc ion, Zn2+. It may be absorbed as a chelating agent, EDTA. Now, the overall functions of zinc. Zinc is an enzyme activator and involved in protein hormone, indole-acetic acid IAA, and RNA DNA synthesis and metabolism, and in ribosome complex stability. Zinc is neither oxidized or reduced in plants. Its functions are based on properties as divalent cation with a strong tendency to form tetrahedral complexes. It is therefore either a metal component of enzymes or acts as a functional, structural, or regulatory factor of a large number of enzymes. Now let us look at the overall functions specifically on enzymes. There's alcohol dehydrogenase, which catalyzes the reduction of the acetaldehyde to ethanol in anaerobic respiration of meristematic cells at root apices. Zinc is actually required there. Sod superoxide dismutase, associated copper zinc sod complex in the chloroplast, which protects chloroplast against photo oxidation. Carbonic anhydrase catalyzes the hydration of carbon dioxide and water into hydrogen carbonate and hydrogen ions. This reaction is used for carbon dioxide storage in the chloroplasts for photosynthesis. RNA polymerase zinc is an essential component of RNA polymerase and ribosomes. In its absence, ribosomes disintegrate and the reconstitution occurs on addition of zinc. RNA disintegration enhances zinc deficiency because zinc is an inhibitor of RNase, an enzyme which hydrolyzes RNA molecules. Protein synthesis is severely reduced in zinc deficient plants. Most visible symptom of the deficiency is stunted growth due to shortening of internodes, rosetting, and drastic decrease in leaf size, little leaf. In cereals, chlorotic bands develop along the midrib, accompanied by red spot-like discoloration, that is in maize. Zinc deficiency, since this element is not easily mobile, apical or young growth is inhibited. However, both young and old leaves can show intravernal chlorosis, pale green, yellow, or even white. Grain and seed yield reduced more than total dry matter production because zinc seems to play a role in fertilization. Pollen grains have a very high zinc content. Now, zinc deficiencies can be induced by high levels of phosphorus, nitrogen, copper, or iron. This is a maize plant showing zinc deficiency. Again, zinc deficiency on tomatoes. Symptoms can include abnormalities in the roots and shoots with a general standard appearance. Internode length and leaf size may be reduced. Leaf edges can be knackered or distorted. Now let us talk of zinc toxicity. Zinc toxicity is associated with the use of sewage sludge with a high zinc content. It causes inhibition of root elongation and chlorosis of young leaves. Increasing soil pH by liming is the most effective method of decreasing zinc availability and toxicity problems. The next micronutrient is copper. Uptake into the plant is in the ionic form copper Cu++, appears to be an active process and can be inhibited by zinc. Copper uptake does not appear to be sensitive to pH changes, as does manganese or boron. The overall function, copper is involved in chlorophyll synthesis with nearly 70% of copper in the leaves found in the chloroplast. It is also a constituent of plastocyanin, a chloroplast protein that is part of photosynthetic electron transport system. We'll later on talk about this plastocyanin. It passes electrons to P700 in the light reaction. We'll talk about this. Copper is also a constituent of several oxidases, enzymes that catalyze oxidation reduction reactions. It may play a role in elemental nitrogen fixation in legumes and in the production of vitamin A. It may also be involved in RNA and DNA synthesis. Now, copper deficiencies or toxicity, impaired lignification is a common copper deficient symptom in higher plants. This is expressed as stunted growth, distortion of young leaves, necrosis of apical meristems, bleaching of young leaves, white tip, and cereal deficient plants is accentuated by high nitrogen application. Copper toxicity. Copper toxicity may induce iron deficiency and chlorosis will occur. Root growth inhibited with enhanced lateral root development. Copper toxicity may be induced by long-term use of copper-containing fungicides or pig and poultry manure with high levels of copper. Other symptoms include overall reduced growth and standing, as well as thickening and darkening of the roots. Copper tubing and fixtures should be avoided in irrigation equipment to reduce the input of unwanted copper. Molybedenum. This element is needed in the smallest amounts of all the mineral elements and is absorbed into the plant in the molybdenate form, MO4 2-. Uptake can be inhibited by sulfate ions and low pH, but enhanced by phosphate ions. Molybedenum deficiency. Molybedenum deficiency affect pollen formation. In maize, tasseling is delayed. Most flowers fail to open and the pollen germination is impaired in the element's absence. In Zimbabwe, maize seed is dressed with molybedenum to prevent these problems. In legumes, molybedenum deficiency is reflected in nitrogen deficient symptoms, chlorosis, and standard growth as a result of impaired nitrogen fixation process. In other plants, small, irregularly shaped leaves or whiptail are exhibited. Since this element is somewhat mobile within the plant, symptoms usually start with the older growth and progress to the young growth. This includes interveinal chlorosis, similar to nitrogen deficiency, mottling, and sometimes marginal scorching or inward capping of the leaves. Well, this is the molybedenum deficiency, where we have necrosis at the edges. Now we're moving on to chlorine. This element is actively taken up by plant roots in chloride form. Now, on the overall functions, chlorine is an activator for enzyme that releases oxygen from water during photosynthesis. It also is involved in respiration. The negatively charged chloride ion also acts as a counter ion to the positively charged cations in the cell. Chloride is involved in regulating turgor pressure and growth of cells and is important in drought resistance. Chloride may also be beneficial in disease prevention, especially promoting healthy growth of plant while creating a root zone and osmotic properties detrimental to pathogens. So chlorine can prevent diseases, and chlorine is also involved in the opening and closing of the stomata, thus controlling gaseous exchange. Deletion symptoms. Since chlorine is mobile within the plant, symptoms appear first on the older growth. Leaves will become chlorotic and finally necrotic, with the leaf area being reduced. Wilting is common and transpiration can be reduced. There is an overall standing of the plant and subsequent dieback. Toxicity symptoms. High amounts of chloride or chlorine produce typical salt stress or salinity effects, including leaf tip or edge, burning, chlorosis, bronzing, and premature leaf drop. Nickel is our last micronutrient, which was recently added to the 7 micronutrients to become 8. Nickel is a component of enzyme urease. It is present in a wide range of plant species. It is a key component of enzymes involved in nitrogen metabolism and biological nitrogen fixation. Nickel deficient soybeans accumulate toxic levels of urea in their leaves. Nitrogen deficiency can cause chlorosis in young leaves of tomatoes. So this could be nickel symptoms, deficient symptoms at the edges of the leaf. So this is the end of the lecture on micronutrients. Please remember that the micronutrients are required in small quantities, but they are very crucial in plant growth. They can also affect human health. That is the end of our lecture.